It's my pleasure and honor to introduce tonight's speaker, Dr. Danielle Zacherl. Danielle is a marine ecologist and professor in the Department of Biological Sciences at California State University, Fullerton. She earned her PhD from UC Santa Barbara and then pursued a postdoctoral research position at UCLA, where I actually met her. Before she joined the faculty at CSU Fullerton. She has initiated several efforts to restore Olympia oysters in Southern California and has been studying the population, biology, and ecology of this native species for nearly a decade. Recently, her interest has expanded to the introduction of non-native Japanese oysters into Southern California bays and estuaries. The title of tonight's talk is, If We Make Their Bed, They Will Lie, Restoration of Native Olympia Oysters in Southern California. Please help me welcome Dr. Danielle Zacherl.
Well, thank you. I am delighted to be here. I especially want to thank the sponsors of this evening. They took me on a really nice dinner to a local seafood restaurant. It was delightful. And I guess I'm here tonight, of course, to talk with you about efforts that we've made to restore native Olympia oysters in Southern California. And in this picture, I'm showing you a recently restored bed from Newport Bay. And if you look really carefully, you can see lots of little Olympia oysters sort of poking up out of the shell and out of the mud. Okay? And my hope is that I'm going to focus on 3 stories from 3 different bays in Southern California, and I'm going to talk about efforts to learn more about the ecology of the species and also to learn more about the best ways to restore the species in Southern California. So I first want to acknowledge the fact that I'm going to present a lot of data generated by my undergraduates and my graduate students today. So all the students that are listed in bold here are students whose data I'm going to be presenting today. And actually, the vast majority of them are undergrads whose data I'm going to present tonight. I'm very proud of them. And then before I get started, I also want to thank my financial supporters, and particularly I want to thank CSU Coast because, of course, they're helping to fund this evening's talks and this series of talks. So, the star of the show is the Olympia oyster, Ostrea lurida. And in this picture here, I am showing you a full-sized adult. And if you look very carefully at the shell, you'll notice that there are recruit oysters, little juvenile oysters that have recruited to the shell. And this is the only oyster species native to the US West Coast. And you may not know that there were previously oyster beds in Southern California. This is something that I talk with folks about a lot, and they're often surprised to hear that oyster beds were present and, in fact, supported a very small industry for a short period of time. And there were sizable oyster beds in Alamitos Bay, Newport Bay, and in San Diego Bay. This species declined pretty much across its range. So the map I'm showing you here is indicating locations where it was historically present. And it declined significantly in the early 1900s due to a combination of overharvest, but also pollution and habitat loss. And habitat loss was in the form of infilling and dredging, development, et cetera. And while there has been restoration underway for a couple of decades in Washington and Oregon, we're really just kind of getting started here in California, and particularly in Southern California. So when the populations declined, they weren't completely decimated. It wasn't like they completely disappeared, but the habitat, the bed habitat, did disappear. So we do, however, or we always did, maintain remnant populations of these oysters. So they weren't living in beds anymore. They weren't bed forming, but what they were doing was living on pier piles and seawalls and pieces of cobble in the habitat, and so they were sort of eking out an existence in that fashion. So what happened in Southern California and across the range of this species isn't unusual. Actually, oyster reefs are one of the most severely impacted habitats on the planet. And a recent paper that came out by Beck et al. In 2011 estimated that 85% of the world's oyster reefs have declined or been completely lost And if you look at this image, you can see that it's really a global phenomenon. The only place where there really are reefs anymore in good condition are a couple of locations in South America. And you can see that along the entire coast of the United States, that our Olympia oyster is either in really poor condition or it's functionally extinct. And when I say functionally extinct, again, I don't necessarily mean it's completely absent. It's just not forming beds anymore or reefs anymore. Okay? And that's too bad, because oyster reefs and oyster beds play a really pivotal role, and they contribute a remarkable number of different ecosystem services to bays and estuaries. The first and perhaps most important one is that they're very gregarious. They like to settle on one another, and they like to form these beautiful reefs or beds when they do that. And if you look, you can see that the reef or the bed that's formed is full of cracks and crevices. And this is habitat that's used by juvenile fishes. It's used by octopus and crabs. It supports a whole community of organisms that otherwise don't have so much shelter. And on the US East Coast, oysters play a really pivotal, it's been documented what a pivotal role they play in providing refuge and food for important fishery species like sea bass, snapper, and flatfishes. And I just wanted you to take a moment and think about whole habitats disappearing from a landscape. So think about Southern California and the iconic habitats that we have here: kelp forests, seagrass beds. And what if they completely disappeared from this landscape? How different life would be in Southern California. And effectively, that's what's happened with oyster beds. In fact, they've disappeared, they've been gone for so long that they're really not in anybody's memory anymore. Most people that I talk to say, I didn't know there were oysters in Southern California. Well, there's oysters. We had beds. But so the other important ecosystem services that oysters perform is they're filter feeders. So they actually improve water quality by filter feeding phytoplankton and bacteria and such out of the water column. And in this picture, I'm showing you that if you put a pile of oysters in an aquarium and you dump a bunch of algae in there, a couple of hours later, you're going to have very, very clear water. And this is really good for eelgrass, of course, because eelgrass are a plant that rely on sunlight penetrating through, our turbid waters, right, to photosynthesize. You might not have known that the endangered California least tern actually build their nests using oyster shell. Think of what a cozy nest that would be. And I'm showing you, just in case you didn't believe it, I'm showing you a couple of pictures of least terns that have done just that. They have gathered up fragments of oyster shell and build their nests out of them. And in fact, some of the restoration efforts that have been focused on this species have focused on delivering oyster shell to those habitats so that the least tern can use its favored material for its nest building. Okay, and then lastly, oyster reefs and oyster beds can play a really important role in stabilizing shorelines. And there's a movement across the entire nation, actually, to implement these more natural techniques for stabilizing sediments and stabilizing banks. And so here I'm just showing you an example of what an estuary might look like if, rather than having a seawall or riprap, we replace that seawall or riprap with natural vegetation and maybe a living breakwater, and that could be made, of course, of an oyster reef or an oyster bed. And that would serve two functions, of course. It could be functional in terms of harvest, right? And it could be functional in terms of offering habitat. I guess that's three functions. But it could also break waves, right? And break wave energy. Okay, but there are some challenges to restoring native Olympia oysters in Southern California. And one of the most giant ones is that we really have very few quantitative data on what natural beds looked like in Southern California. We don't know how extensive they were for the most part, except for a few mentions in the late 1800s about, for example, in Alamitos Bay, having a bed that extended half a mile long. But outside of that, we don't know how dense the oysters were on those beds. We don't know how tall the beds grew, right? And, but just to give you an idea how extensive an oyster bed can be, so far I've just showed you tiny little clumps, right? But this is a picture of the non-native Japanese oyster which has invaded Germany. And you can see that it is completely, I mean, it's just a landscape of oyster, okay? And here's a picture of native Crass Ostrea virginica in Georgia. And here, again, you can see all that wonderful rugosity, and you can see how extensive a bed could be. And you could imagine how much habitat that could offer to estuarine organisms. And the last thing that I want to talk about before I start talking about our research is the fact that oysters, we don't just have evidence from the 1800s and the 1900s that they were here. We actually know from fossil deposits that they've had a presence in Southern California for at least a million years, okay, since the Pleistocene. And so here I'm just showing you a picture from Newport Bay. This is one of my former undergraduates. His name is John Berryman. He is holding a ruler up to some oyster shell that's deposited into the cliffs. And we worked with some geologists at Cal State Fullerton to date that shell, and they came up with an age of about 600 years old. And if you go inland into Fullerton, where I'm from, actually you can find deposits that extend back to the Pleistocene. And in San Diego Bay, it's been very well documented, their historic presence. The San Diego Natural History Museum has collections all over the place because every development project that happens in downtown San Diego uncovers oyster beds or clam beds. And so they have just extensive collections. And just one thing I'll mention is that a paper that came out in the 1881 noted that in La Punta on the south side of the bay that there were definitely enough oysters there to harvest, but they also noticed that they had a really unusual coppery flavor. And this is really interesting because up in Washington, where they've been most successfully restored, there is actually now a market, a specialty sort of niche market for this wonderful oyster because it's said to have a particularly umami flavor and to be just quite lovely to eat if you're into eating oysters. But there was never really a major industry or a major harvest here in Southern California, as far as we know from the literature. Okay, and then just to show you again the extent of those deposits I was talking about. So all of these green dots on the map of San Diego Bay are locations, many of them just right in downtown San Diego. And this is just a scratch of the surface, like what I could put on a map, in a half an hour's time, collections that have been found of oysters present all over the place. And then I started to document their presence in the literature as well with these different colored markers. So in other words, these are studies that came out in these years that indicated that oysters were present in San Diego Bay. So they've been here for a while. Okay, so one of those challenges I mentioned was that we just don't really know what a native oyster bed should look like. So what are we restoring to, right? What's our mark going to be when we know we've succeeded in restoring them? But a second challenge is that we have started to see an uptick in the presence of a non-native species, Crass Ostrea gigas. This is the Japanese oyster. It's also sometimes called the Pacific oyster. For those of you who are oyster aficionados, it's probably the most commonly cultivated oyster species. The picture that I showed you in Germany indicates just how aggressive it can be as an introduced species. It's not always that way. It has had many failed introductions, and in Southern California, we're kind of waiting with bated breath to see what's going to happen. But here's a picture of a full-grown Crass Ostrea gigas, the Japanese oyster, with several full-grown adult lurida that have settled onto the shell. So it's not really clear that they're in any kind of direct competition. In fact, they live pretty happily among one another, right? But they are a species, that's present, that's a non-native, and so that raises the scepter of a real problem, which is if we build a bunch of shell beds and we want to recruit just the native oyster, how are we going to prevent, right, the Japanese oyster from setting on that same habitat that we've set out for Olympia oysters? And that's been a real challenge with permitting agencies to work with them to convince them that maybe restoring one species is not going to introduce a new problem, okay, to Southern California. And then just to give you an idea what a Crass Ostrea gigas is truly, it's a beautiful organism. And when it's When it's not old and worn down and it's a juvenile, it has just these wonderful fluting on the shell. And so here I'm just showing you a recruit. They grow so fast and they look so pretty. Yeah, so that's what that's showing you. Okay, and then one more problem I want to talk about, and that is that, you know, on the East Coast, a lot of work has been done on what's the most efficient way to restore Crassostrea virginica, the East Coast oyster, but none of that kind of scientific work has really happened until very recently on our coast. And so when we started working with this species to restore it back in 2010, we really didn't know what technique was going to work best. Well, in general, the easiest way to restore an oyster and to try to restore the habitat of an oyster bed habitat is to just simply take a bunch of dead oyster shell from a local oyster farm and dump it out on the mud. And then that is prime real estate. And we all know in Southern California how important it is to make a smart real estate decision. And these oysters have got one chance, because once they make that decision, they're cemented for life, right? And so they've moved in and that's it. No more moving out of the neighborhood. Okay, and so the easiest way to do it is to simply dump the shell on the bed or on the mud. But sometimes we bag the shell to keep it consolidated so it doesn't just float away, right? And we didn't know, again, it relates back to the stuff I said in the beginning about how little information we have about this species when it was in its prime. We don't really know how big to build the beds. Should we build them 3 feet tall? Should we build just to put a dusting of shell on the mud flat? We don't really know what works best. And it also begs the question, are we trying to restore back to what was once present? Do we want the beds to look like historic beds? Or rather, do we want to build beds that are going to help Olympia oysters perform the best for us? Right? And so those are two really very different kinds of questions in restoration. And so we set out to ask some questions like, which bed height might be best? How thickly are we going to apply this oyster shell to the mudflat? And we also wanted to know, we're going to put them in the intertidal. Well, what tidal elevation should we put them in? Should we put them in the low tide zone? Should we put them underwater? Should we put them up high? You know, there was really no information about what was going to work best. So in 2010, we started a restoration project. This was a pilot study in Upper Newport Bay, just north of Highway 1. Those of you who are familiar with Newport Bay may have been to this site. It's Castaway's Mudflat. It's a lovely little spot. This is a picture of a Castaway's Mudflat, and you can see, you know, that's what I said. It's a mud flat, you know, it's mud. There's no hard habitat, structured habitat for oysters to settle. You might notice in the distance there is some cobble up here, and some of this is natural shale, and some of this is introduced like concrete, human-introduced concrete. But for the most part, at the tidal elevation where the Olympia oyster resides, it's really just 100% mud, okay? And so we set out to answer some research questions. We first, again, wanted to know, did it matter how tall the bed was or how thickly we applied the shell? And so we applied shell in 2 thicknesses. We applied it really thin, about 4 centimeters thick, and then about 12 centimeters thick. We built 25 beds in all on this beach. And each one of those beds measured about 2 meters by 2 meters. And we had different treatments. We had thick beds and thin beds. And then we had some beds where we just applied the shell loose to the mudflat, and some beds where we bagged it up to make it easier to transport and also to consolidate it more and make it harder to lose the shell. Okay, so in all, 25, and then we had some plots where we did nothing at all to the mudflat. We just left it like a big old mudflat, okay, with no shell at all. And so in all, 25 experimental plots with 5 treatments, and I'm showing you the 5 treatments here. And in a second, I'm going to show you a couple of data slides. And so I want you to be familiar with the terms I'm using. I'm going to call these thin beds, and these are going to be thick beds. And we either bagged the shell or we applied loose shell. And I just want to make the point that we're not putting live oysters onto the mudflat. What we're putting onto the mudflat is dead shell. And the hope is that we're going to get that remnant population that's just been eking out its existence, well, they're going to produce young oysters that are going to settle onto our beds of shell, right? And so that's the plan, and we wanted to know which one's going to be the best. All right, well, so we monitored these beds. We built them in June of 2010, and we monitored them for 2 years. Basically, every 6 months, we would go out to those beds, and we would excavate a piece of the bed, and just to see how the oysters were doing. How were they growing? How many were recruiting to the site, et cetera. And now, what I'd like to do is show you I want to show you, first of all, what a bed looked like. So remember, I just showed you that mud flat with nothing on it. Well, so this is that dead Crass Ostrea gigas shell that we set down. And of course, all these things poking out here, these are live Olympia oysters. And this is, this picture that I'm showing you here is actually after just 6 months. So after just 6 months. And after 2 years, the densities on these beds were 26 times higher than the starting densities at that site. And they were double the average densities elsewhere in the bay. Okay, so that was a significant finding. And interestingly, we didn't recruit a single non-native oyster. It was all just Olympia oysters. That was a little mystifying to us, but we've solved that mystery, we think. Okay, and I'll get to that. And we basically reached, oh, and incidentally, I want to give a shout out to 2 just phenomenal undergrads who actually, together, the 3 of us just had this paper accepted this week, the data that I'm about to show you. So we're very excited to publish this work. This is Shannon Crossan and Andrea Moreno, who did just so much work for me over several years. But so in any case, the general conclusion we came was that it didn't matter how you built your bed. You add shell to the mud flat and you're going to get oysters. And it doesn't really matter what technique you use. And so these data that I'm going to show you next kind of bring that point home. So on this, I'm going to walk you slowly through these. I'll be showing you a couple. On the x-axis, we have the treatment group. So this is the type of bed that we built. Remember, we had thin beds and thick beds. We had loose shell and we had bagged shell. Okay? And on this axis, I'm showing you how many adults per meter squared. How many adults per meter squared? Well, a meter squared is about this by this, right? And let's see. And so what you can see is that over time, these are data from once a year we took in June, okay? You can see that the control plot started with very few oysters and they effectively had none by the end, okay? Whereas no matter what kind of shell we laid down, they almost all started with no oysters and they all gained similar amounts of shell. Now, if you haven't ever seen these, we call these error bars, these bars right here, and those are an indication of how noisy the data are. And what that means is, remember we had 5 reefs of each type, and we kind of wanted to average how many oysters there were on a thick reef that was bagged with bagged shell. And so this gives you some idea that some of those reefs that were of that design had few oysters and some had a lot. Okay, and this isn't exactly the range of the data, but it gives you an estimate of how confident we are in the mean. And so you can see that the means, we know that there's quite a few more than the control, right? But relative to one another, because there's so much overlap in these bars, that is a sort of a coarse way to eyeball whether there's any difference among these groups. So you should be looking at those error bars for some indication of how noisy the data are and then if there's a difference among the groups. And so what this graph tells us, again, is that there's no difference. It doesn't matter how you put the shell out. You put shell out, you get oysters. It's great. We were very excited about this outcome. However, when we looked more closely, we didn't just look at adults. We also looked at recruits. And recruits are, we were calling them anything that was microscopic that we couldn't see with our naked eye, but we knew they were more than several weeks old. We would call those recruits. And you can see that in the first year, we didn't get a tremendous amount of recruitment actually onto those beds. And there really wasn't a difference among the groups. But by the second year, man, was there a difference. We just had so many recruits on those thick beds, and they hadn't grown out to adult yet. So we don't really know the fate of those recruits. But this is a signal that's indicating that something about those thick beds was really appealing, right, to those recruit oysters. And so, of course, if we had to hypothesize what would happen in the future, we would say the next year we probably would have found a lot more oysters on those thick beds. But funding ran out, and we moved on to another project. So I don't know what happened after that. But I have gone back to that site, and there are still lots of oysters on that mudflat. Okay, then the last piece of data I want to show you involves the integrity of the beds. So one of the dirty little secrets about doing oyster restoration is that you put a bunch of shell down, and if the oysters don't build it up fast enough, the beds are going to get buried. They're going to get buried by sediments, or they're going to get carried away by the currents, or a boat wake will come by and knock some of the shell off and it drifts away. Okay? And so you have to be really conscious of the integrity of the bed. And these data sort of further indicate to us, so we're looking at how much shell was covering the mudflat. And what you can see is that the thick beds, the blue here, they didn't lose very much shell cover at all. They started with 100% shell cover because we laid a lot of shell down on that mudflat. Okay, but over time they kind of stabilized, and by the end of the study they were at about 80% cover of shell. So there was still really a bed there present. Okay. On the other hand, the beds that we built that were built with very little shell, they lost quite a bit more in terms of the percent cover. And we also saw that some of that shell drifted off of our experimental beds and onto our control beds. Okay, but nonetheless, even though we did get some shell building up on our control beds, we never saw oysters recruiting to those beds after that first year, which is pretty interesting. Okay, and so just to sum up that part of the talk and what we learned in Newport Bay in that first 2 years of work was that bed thickness, by the end of 2 years, it didn't matter in terms of how many adults we had recruited, but we think because the beds held their integrity better, and because that last year they got so many recruit juveniles, we think that, that maybe bed thickness does matter. But we never saw, you might notice in those data slides that I showed you, that we never saw any difference between whether they were bagged or unbagged, right? It never mattered whether they were bagged or unbagged. And so, and this is a good example where, you know, this is our loose and our bagged, and they're just completely overlapping, okay? So no difference. So we say that the consolidation of the shell, or applying them loose or not, it doesn't matter. And so on the one hand, bagging them is a great way to get the public involved, right? And get them to help you build the beds. And they can bag the shell with you, and it's an awful lot of fun. We actually hosted several of those events. It was a great way to get the public involved in this project, but it costs a lot of money to buy those bags, and we bag them in biodegradable jute. And so you know, the savings from that might make it that we just would like to use, you know, to apply loose shell. So we moved up north, and so the recommendation that we came out of this study was future restoration projects should apply shell thickly. And we took our own advice, and we moved to Alamitos Bay, and we built another bed. And before I take you to Alamitos Bay, though, I just want to make that point. Remember I said that oyster reefs are such important habitat suppliers? We have a whole other portion of our study that I'm not going to talk about tonight that has to do with what great habitat they provided. And we looked at community diversity on these beds to see how the community changed when we added shell. And we had so many cool things recruit to our beds, like scallops. We had just a giant number of scallops recruit to the beds. Sponges, tunicates, amphipods, copepods, isopods, crabs, really cool octopus we would see out there all the time, mussels and bryozoans, so all sorts of organisms were recruiting to our beds. That's another story for another day, but we were pretty excited about that outcome as well. Okay, so we moved up to Alamitos Bay, and we decided we were going to build another bed. Taking our own advice, we built it thickly, this time 8 centimeters thick, and we built one bed, and it measured 2 meters by 30 meters long. Okay, and this was not an experiment. It was really more of a demo project to get the public. It was a public awareness campaign and an outreach education campaign, and so we really just wanted to build one bed. We still, however, sampled the heck out of it because we had all sorts of monitoring requirements because of permitting restrictions. When you build anything, it turns out, incidentally, in Southern California, those of you who have shoreline property will appreciate this, it takes a lot of permits to put anything out into a natural habitat, including a restoration project. And so that process was a really, really long process, getting the project permitted. So in any case, the other thing I want to say about this, though, is that it wasn't really set up like a scientific study, but we learned a lot from this location, which I'm going to share with you right now. But I want to make the point that if you look carefully, you can see that there's eelgrass habitat just below this oyster bed. And that raised a really interesting question in our minds, because many of you may know that eelgrass restoration is a really hot topic in Southern California now. It's happening everywhere. And there was a lot of concern by the permitting agencies when we set up these projects. Were the oysters going to hurt the eelgrass? Right? And so really there was no data. There were no data out there that could help inform us. And so part of the monitoring we did was a thesis project for one of my master's students, Sarah Briley. I don't have time to talk about it today, but she did a whole project on how the eelgrass responded to this bed. Sarah Briley has now moved on to OC Coastkeeper. She's the restoration coordinator. Super proud of her, and I'll be talking about a future study that she actually is heading up at the end of this talk. Okay. So this project that we did in Alamitos Bay, this time we partnered up with a whole bunch of people, and I would just want to mention them really quickly. Christine Whitcraft, I don't know, do we have any Cal State Long Beach students here? Yeah, have any of you all have Christine Whitcraft as an instructor? I hear dead silence. I guess not. Well, she's wonderful. You guys sign up for her classes. She's great. Okay, so in any case, she really helped us work on community diversity studies, okay, and talking about like who's recruiting to the bed. She really helped us key out those species and figure out who was it that was living on the beds. We had lots of education and outreach efforts that were sponsored by Orange County Coastkeeper. And then lastly, we teamed up with KZO Education and they developed a website for us as part of this project. Okay, we also had about $89,000 in funding from the NOAA Restoration Center and the State Coastal Conservancy, and they've been huge supporters of this research, so just a real quick thanks to them as well. So here's what it looks like when you get a bunch of volunteers out on the mudflat at 5 a.m. and ask them to haul 6,000 pounds of shell down the beach. So here we are, we're in a chain gang, and actually this woman right here, Laura Ellsbury, is out in the audience tonight. I saw her earlier tonight. So what we would do is we basically formed a chain gang on the mudflat. Now, anybody here ever walked on a mudflat before? Okay, yeah. You know what happens when you go onto the mudflat? If it's firm, you're in good shape, right? But if it's not, you can sink a good long way down. And that sinking, it's not only hard for you to handle, but it's actually kind of hard for the mudflat to handle. It's a lot of gouging to the mudflat, right? So what we did was we laid down these crates to sort of make it like we were birds, shorebirds, right? And thus, rather than gouging our boots into the mud and sinking down to our knees, we just would sink down about this much in those crates. And so we would set people up in a human chain gang up down the whole length of the bed, and we would hand down crates of oyster shell, and we basically buried our footprints. So we worked on the outside, and as we worked our way back, we would lift a crate and then cover our footprints with the shell. And in this way, we had, really no visible impact at all from the actual construction project. There was no gouging or messed up mudflat as a result of that. And so we monitored this bed also for 2 years, just like the Newport Bay study. And one of the things that was interesting to us, though, we were really concerned about doing this project in Alamitos Bay because it was supposed to be a demo project for the public. And you don't want to set up a demo project and have it fail because that's kind of embarrassing. And so, but besides that too, you want people to believe this could really happen. Like we could really restore oysters in Southern California. We were really nervous about doing this project there because work by my student, Nicole Tronsky, she's been surveying oysters in multiple bays and estuaries for years now. And she had built this database that showed that our native Olympia oyster, Ostrea lurida, in blue here, in Newport Bay, they were in pretty good density, right? So this works out, this is per quarter meter squared. So per meter squared, this is like 25 oysters per meter squared, right? And that's a decent amount. But the non-native really wasn't very low densities in Newport Bay. Okay? Well, in Alamitos Bay, the native and non-native are about equal densities. And so we were worried there just wasn't enough remnant population to seed our bed. We'd put all that dead oyster shell out there and nothing would come, right? So the second challenge that we had in Alamitos Bay that remember I said we didn't lose much shell in Newport Bay? Well, in Alamitos Bay, we lost a lot of shell. We, in fact, pretty much our bed disappeared after the first year. It was a little bit terrifying to go back out there as a researcher and think, oh, we put so much money into this, and our shell bed is all gone now. The shell cover dropped dramatically that first year. And so we came back out, and what we did was we noticed that there were these natural channels that kind of formed with bed in pretty good shape. And then we'd get a channel where everything got buried, and then we'd have another little clump of bed in pretty good shape. So we decided we were going to use an adaptive management strategy, and we were going to apply more shell, but we were going to mimic what we saw happening at the site already. Okay, and that's what we did. We went out there, and so this is the old original bed, and you can see it looks pretty much like a mud flat. And then here were the new shell beds that we laid down, and we laid them down in 3 mini beds instead of 1 long bed. And here are my students on their crates doing their survey work. Okay, and so here's our new bed. And so then Oh, and then incidentally, I want to mention that in this design, we were also monitoring the mudflat in the distance here. So we were monitoring, this is about a 30-meter long stretch of oyster reef, and we were also monitoring a 30-meter long stretch of mudflat because we obviously wanted to have some reference, right, to what we were building. And so these data were generated by my student, Cristina Fuentes, a wonderful, wonderful student. And she monitored for 2 years. And in this case, when we started the study, well, we weren't monitoring the new bed because we didn't intend to build it, right? So we have no data for that portion of the mudflat. We had no oysters at all at this site when we started. Look at what happened at the end of 2 years. We had just this giant amount of oysters, just a giant amount, 62 times reference densities from other local locations. That's a giant amount. I mean, it was infinite increase relative to the mudflat that we started at, but relative to the densities elsewhere in the bay, 62 times the densities. These densities that we were getting here were as high a densities as exist across the range of this species, even in places like Washington and British Columbia, where they've been restoring this species for decades. So we were super, super excited about this result. There was a caveat, however. Remember how in Newport Bay we didn't recruit a single non-native oyster? Well, on these beds, we recruited non-native oysters. They were about 2 to 3 times the local densities otherwise. And so here you can see that if you look at, we didn't recruit them onto the control plot, so it wasn't just the time, right? It didn't happen on the control, it happened where we applied the shell. We got, on both the old bed and the new bed, we got Crass Ostrea gigas recruiting. And the question, of course, that begged the question, like, why here but not in Newport Bay? Well, there was one giant difference between the two locations, and that was that we built the beds in Newport Bay at a lower tidal elevation. Not much lower, just like a foot lower, just like this much, okay? But that made all the difference, we think. And so we have a hypothesis, and it's based on work that my student Thomas Parker has done, that there is a really strong zonation pattern for these two species, where Crass Ostrea gigas lives up high in the intertidal, above the mussel zone, okay? And Ostrea lurida lives really low in the intertidal. And so here on this graph, I'm showing you on the x-axis the tidal height in meters, okay? And as you move up towards terrestrial habitat, okay? So this is the low tide area, okay? The low intertidal, this is the upper intertidal. Okay, and so for those of you who've been out in the intertidal, this might be equivalent to like the barnacle zone, and this might be equivalent to the mussel zone, okay? And what you can see is in 2 bays where we did survey work, in Alamitos Bay and in Huntington Harbor, the trend was that we would have higher densities of Olympia oysters if we were lower in the intertidal, below the mussel zone, and they would decline radically as we moved up into the intertidal. And the exact opposite thing would happen for the Japanese oysters. They would increase, okay, as we moved up higher. And if I map on the height, the tidal height of the beds, I want to show you that in Newport Bay, the beds, you know, they were equivalent to a place where we really weren't seeing Crass Ostrea gigas, okay? Whereas the beds we built in Alamitos Bay were at those tidal elevations where we would start to see a decline in natives and start to see an increase in non-natives, okay? And so in Alamitos Bay, remember I said we had our original bed and then below it we built our new bed, and even just that difference in tidal elevation, right? And so let me show you what that difference looks like again. So it's a very shallow slope, but just even that difference, when we sampled on this side of the beds, we had about a 44-to-1 ratio of natives to non-natives. And when we sampled on this side of the beds, about a 7-to-1 ratio of natives to non-natives. So that's a giant shift. And I just want to show you a picture of what that zonation pattern looks like. So we're moving down to San Diego Bay now, okay? And this is a chain link fence. And remember I was talking about the mussel zone? Well, the mussel zone is this black zone right here. That's a solid wall of mussels. And you can see above the mussel zone, this is a solid wall of Japanese oysters, Crass Ostrea gigas, okay? And below, and intermixed, and because they're so small and they're covered with mud, you can't see them, but there's just a whole bunch of native oysters down there, okay? And when we looked at that pattern of Density, again, as a function of tidal height. So again, here we have tidal height, and each one of these data points represents a quadrat that we popped onto the wall, and we counted how many native and non-native oysters there were. And you can see the same pattern existed, that we had lots of native oysters down low, right? And they would decline radically as we got up high. And lots of non-natives, or not very many non-natives down low, they would peak right at this point. And then when we got into basically terrestrial habitat, they would decline again radically. Okay, so they haven't actually succeeded in making the jump from seawater to land, but they're getting close. They're getting real close. They're living right there on the edge. This is really the splash zone here. It's remarkable. This species is a remarkable, remarkable survivor. Okay, so I just want to make the point that in San Diego Bay, there's a lot of native oysters there. They're doing really well. It's a very healthy, natural remnant population. And you can see, if you go to certain places in San Diego Bay, there are little patches of bed. They're very small, so this might be, a foot across, right? But there are patches of beds like that. But here are some plates these are what we call, in my field, we call these settling plates. These are setting plates. We put them out on the mud flat. We suspend them upside down, right? And we suspend them upside down and we leave them out for a period of a couple of months. And when we pull them back into the lab, we can put them right under the microscope, right? And we can count how many oysters we have per unit area. And so here I'm showing you a setting plate that was in a location in San Diego Bay, and it's just covered with native Olympia oysters. But if you look really carefully, and you remember what I told you about those non-natives being so pretty, you can see that there's non-native oysters there as well. And what's so interesting about Olympia oysters is they like to settle on the undersides of things. And so this side, this is the same shell plate, just flipped. It's a tile. It's a tile that you would buy at a hardware store. And incidentally, I have been audited before by my school for buying boxes of tiles, Diablo red tiles. I've had them calling me saying, why are you buying hundreds of dollars worth of tiles with your funding? And this is why, I'm using it scientifically, okay? And so this is the same tile, and this is the side that we suspended above the mud flat, right? And so this is the side that was the underside. And this is the side that was the top side. And look at these are non-native Japanese oysters here that have settled on the top side. No Olympia oysters on the top side, just Japanese. And they had this remarkable survival rate. So we did a survival and growth study, and the Olympia oysters just get hammered. It's like 10% of them survive for 3 months. The Japanese oysters, 60% of them, 50% of them survive. I mean, they're just survivors. Their densities in San Diego Bay are 7 times greater than elsewhere in Southern California, and their survival rates, again, greater than 50%. And there's been this really notable uptick in the presence of juveniles. And that's happening not just in San Diego Bay, but also in Alamitos Bay and in Newport Bay. So we're seeing that all throughout Southern California, this uptick in these Japanese oysters. So just to conclude really quickly, some of the things I talked about, we've figured out that technique to a certain extent matters, right? And I said, after 2 years, it didn't matter what kind of technique we used. We got the same number of adults. But in terms of the integrity of the bed of a long-term project, we think that applying shell in a thicker manner, is going to result in the long term. It's going to maintain its integrity longer than a thinner bed. And we definitely think we've demonstrated, although we're going to do it now in a manipulated study, we think that tidal elevation matters. That if you're going to build a bed, and you're concerned about who's going to move into the neighborhood, build your bed low, right, at a low tidal elevation. And if you build them above a tidal elevation of about 0.2 meters, which is about the mussel zone, then you're going to encourage non-natives to recruit to it. And then lastly, I mentioned really briefly that my graduate student, Sarah Briley, does work on oysters and eelgrass. And we think that that is really an important consideration. It's on everybody's radar who's doing restoration in Southern California. Everybody wants to know how they're going to interact. Are they friends? Are they foes? Are they going to be compatible in terms of restoration effort? And so one of the most exciting future plans, I'm hoping to get involved in 2 projects that are upcoming in Southern California, and they're both living shorelines projects. Okay, and one of them is in Newport Bay, where my student Sarah Briley, who's now the restoration coordinator, she has just gotten a ton of money from the State Coastal Conservancy to restore oysters and eelgrass simultaneously in an experimental design that allows her to look at restoring oysters by themselves, restoring oysters and eelgrass together, and restoring eelgrass by themselves. And we can look at the efficacy of each method and see what's best for each habitat type. The next project that I'm hoping to be involved with is with the Port of San Diego. And they're really big on this living shorelines thing. They would like to restore oyster beds in San Diego Bay, and they're talking about building sizable beds in a formation or a reformation that would reduce wave energy and stabilize shorelines. And so we're working with an engineer to figure out the best configurations, what kinds of designs would work the best. Clearly, building my 30-meter long bed in terms of deposits of sediments onto the bed was not the right idea. So we're working with sort of smaller bed designs and figuring out, how far spaced apart do they have to be, how tall do they have to be, and at what tidal elevation do they have to be to work to reduce wave forces to our shorelines, but also to restore Olympia oysters. And then, of course, I'm really interested. I think Julianne mentioned in the beginning of my talk that I'm really interested in understanding the interactions between native and non-native oysters and understanding what creates that really strong zonation pattern. Okay, and so this last picture I'm going to show you here is just another, it's sort of a leaving you with an image of restored oysters in Alamitos Bay. And so again, these are the giant Ostrea gigas shells just covered with Olympia oysters. And then here are Olympia oysters living very happily on a bed in Newport Bay. And once again, I just want to acknowledge, my students who did such wonderful work. And I'm happy to take any questions you have.
So questions for Dr. Zacherl?
Yeah. Oh, you want to mediate or shall I? Okay. Yeah. Sure.
You didn't seed any of these oysters. That's just
No. So far, once again, so far what we did was we put out dead shell, and we relied on the remnant populations to seed the shell. Now, we have talked about the concept of seeding as a way to boost, to get it started. There is just a little bit, it's a little bit more risky. You have to really think hard about the genetic population structure of the stock you're introducing into the bay. And so it is a step that is used most often when there's not a natural population that could seed the bed, but it could be a solution for us as well to get things jump-started. Yeah, did you want to follow up or no? Yeah, that's it, okay, yeah. Hi.
Hi, are the natives edible, and do they have the coppery taste?
Yeah, well, so they have I don't taste something metallic, I taste umami, so I taste savory. And that's what they're sort of noted for in Washington. But it has been stated that you grow them in different bays, they take on different tastes. And maybe something about South Bay San Diego is going to produce an oyster that's coppery, right, compared to an oyster that's grown up in Washington State. Yeah, yeah. And it's a niche market up in Washington. So there's one shellfish farm that I know of. It's called Taylor Shellfish. And they actually they cultivate Olympia oysters for sale. And the oyster aficionados apparently are crazy about them. So but they're quite small. I think I showed you in the very beginning of my talk. In fact, I'll show you again, a little plate of Olympia oysters. And that's quite a different image than, if you go get some Japanese oysters. Let's see here. Put this back up into slide view. So here's a plate of little Olympia oysters So for somebody like me, I'm a little bit intimidated by oysters. I don't know about you, but I am. And so it's easier for me to eat the little ones. I'm less intimidated by them. So yeah, okay.
I have a question. You have this map here of all the historic locations.
Sure.
Have you found out if there are remnant populations in those areas as well, so that natural recruitment to beds that you place is possible, or if any implementation of larvae is needed?
Yeah, so actually, I didn't get to talk about that, but I will for a second. The way that I got started studying Olympia oysters is I had a student who was really interested in she wanted to do a study that would take her across a broad geographic range. And we had heard some things about the Olympia oyster. This was like 11 years ago. And, maybe we should work on the Olympia oyster. That sounds like a lot of fun. And so she actually traveled. This is not only a map of historic locations. This is a map of every location that she visited and resampled. And she found oysters in every one of these locations and measured their densities to understand their current population status. And there was only one location where she didn't find them, and that was Morro Bay. And it's so interesting because Morro Bay has an oyster fishery there. Yeah, I don't understand. I mean, it's isolated. It's an isolated bay, and so it must have been locally, it must have gone locally extinct, and it just somehow we haven't seen it reintroduced into that bay. Yeah, so that's a little bit of a mystery. Yeah.
I just wondered, for those of us that love oysters,
Sure.
you know, as we buy our seafood, I always try and buy the wild-caught salmon. So, but recently we saw, like, it sounded like not all farmed are bad, obviously.
No.
It's sustainable. So how do we know whether what we're buying is the best for the environment? If you have recommendations of which ones we should avoid or...
I don't necessarily, but I will say this. So Carlsbad Aqua Farm is down in Carlsbad, and they grow mussels and oysters, and they have talked about trying to create a market for the Olympia oyster in Southern California. That's on their radar, and that's something that they're thinking about. And so that's something I of course, I'm always really excited about that idea that, you know, we could bring this native back. But it will, to be honest, it will never be the industry choice for a fishery species because it's slower growing and its maximum size is so small that it's not really going to ever replace Pacific Japanese oysters. Not necessarily. I mean, in theory, these oyster farms are concerned about what they call biosecurity, which is, and they're often producing triploid oysters, and those are oysters that they've given an extra set of chromosomes, basically, so they cannot reproduce. So they can produce them in the hatchery, grow them out in the bays, and because they're these triploid strain, they, in theory, cannot locally reproduce. Now, on the other hand, it sort of begs the question, where did the oysters come from that are in our Southern California bays and estuaries? But there are lots of potential answers to that question. One of them is, sure, they escaped a shellfish farm. A second is, you know, they were introduced by ballast water or by some drift thing, some drift phenomenon, or they came in on boats. They grew up on the bottom of boats and were transported across the ocean, and that could have been their mode of introduction as well. So lots of possibilities, yeah. Yes, ma'am.
In this day, I never heard two words used by climate change, whether there's any climate change, whether there's any problem.
Yeah, well, so it's interesting, actually. The whole Living Shoreline initiative I was talking about is also aimed at helping address climate change issues. For example, sea level rise. The thinking is that if you have a living shoreline, the living shoreline can migrate with sea level rise much better than an implemented, human-introduced structure that you'd have to break down and rebuild again. And so one of the ideas is, so it is of concern. It's on the radar of, for example, the Port of San Diego, and I'm sure the Port of LA is thinking about this as an issue as well. This issue of, what are we going to do with our ports, right, as sea level rise becomes a reality. And so whether you buy global climate change is happening or not, cities are starting to plan around it and thinking really hard about it. Yeah. So does that address it? Yeah.
Two questions.
Yeah,
the
sure.
First one is, did you use the variable of temperature at all, the water temperature? Because it does make a difference between Sitka, Morro Bay, and Southern California.
Oh, absolutely, yeah. So we have never manipulated temperature for this species in any way. Oh, yeah, I will. I'm sorry, thank you. The question was, have we ever looked at temperature as an issue with this species? Is that? Okay, and the answer is we have never, and she made the comment that when we sampled across that giant tidal range, the locations in Alaska are going to obviously be quite a bit colder, right, than they are down in Southern California. And so I have a few things to say about that, actually. One of the interesting things is that with latitude, sometimes the warmest events can happen further north because of the timing of the low tides, which are in the summertime up north. Am I right about that? I'm saying that right, Laura? Yeah, up north, they have summertime low tides in the middle of the day-ish. And down south here, we have our low tides in the middle of the day. They happen during wintertime, okay? And but no, we haven't looked at that, except I will make a comment, and that is that in Washington, the Olympia oyster really doesn't live in intertidal. It lives in the subtidal. It lives underwater all the time, and so it's not exposed with the tides. And the thinking is that that's primarily because in the winter they get freezes sometimes, and that would kill off oysters that had set up high. And so it does affect their tidal elevation. Yeah, thanks for that question. Sure, well, yeah, sure, yeah, absolutely.
And the other question I have is, working with a conservation project in Long Beach with Colorado Lagoon, has there been any chance of anyone thinking that they might do, since we're doing eelgrass, introducing it again, has there been anybody that wants to do something in regard to the oysters and do a project there?
Yeah, so the question was, in Colorado Lagoon, there's all kinds of restoration activities ongoing, and has there been any talk of restoring oysters into Colorado Lagoon? And the answer is yes, there has been talk of restoring oysters into Colorado Lagoon, though we're very uncertain about how well they'll do until the flushing is improved in Colorado Lagoon. I know that's on, maybe many of you are local, so you probably know there's a plan to improve the flushing of the Colorado Lagoon, and at that point, that could make it a really nice habitat for oysters.
Because it would seem that the oysters would be advantageous for the purification of that water, since it is the situation that it is.
Yeah, so the comment was that it would seem that if you put oysters in Colorado Lagoon, it could really help the water quality. And the answer is absolutely, yeah. I mean, it could. The issue, of course, is that the oysters really they like the main channels of bays because there's a lot of flushing. And that's where they're going to grow the fastest and do the best. But it is an interesting idea that you could use them to mitigate a particular location in terms of water quality.
Oh, one more question.
Sure.
We'll get to the last one.
Yes.
What are possible negatives of having oysters reintroduced? And what about oyster poachers?
Okay, so the question was, what are the negatives of reintroducing oysters, and what about oyster poachers? And I want to take the oyster poachers question first because I have a sort of hilarious story to tell. And that is, one day we were Nicole Tronsky was the one who did all the survey work in the Southern California bays. I showed you the densities of the native and non-native oysters. We were doing survey work on a riprap shoulder in Newport Bay one day, and this guy came down and he's like, "What are you doing?" And we said, "Well, we're counting oysters." And he said, "Well, there's oysters down here? I didn't know there were oysters here." And I pointed to a rock, and it happened to be a Japanese oyster, because of course they're the ones that are up high, right? They're up high and they're really big. And so I pointed to the Japanese oyster and I said, "Look, this is a non-native Japanese oyster, and you can't see them, but lower down there are natives." We didn't finish our survey work that day. You know where the story's going? We came back the next day. There were no non-natives anywhere in the site. He had come and just cleaned them off the rocks. Now, he didn't ask me any questions about the water quality, but I would not be eating oysters without, depurating them first. So when you grow oysters in Agua Hedionda, when Carlsbad Aqua Farm grows them, they take them out of the bay for several days, and they filter, they let them live in filtered seawater so that they can get rid of all their fecal waste, right? And they can, and all that bacteria, that local bacteria that they've consumed is all gone. So yeah, so that's it. But then to get to your second and more serious question of, is there any potential negative effect? Well, yeah, some of the concerns, of course, again, are we don't want to encourage non-natives, or at least the permitting agencies don't, and I don't particularly want to encourage the introduction of non-natives. And so there's so little known about who's going to live on our beds that we really don't know whether they're benefiting a native community more or a non-native community more. And remember that when we're building our beds, we're building with Ostrea gigas shell, which is a very large shell. It probably doesn't form the same kind of reef as the little Olympia oyster shells would form. And so we don't really, and even though our shell is dead, it's been out of water for months and months, it's been inspected by Cal Fish and Game, so we know we're not introducing any animals with the shell. It's just who's going to recruit to it, right? Who's going to recruit to it? And so that's a big question. And of course, the second big one is we still don't know whether oysters and eelgrass are compatible. We think they are, because they've historically resided together in these bays for thousands of years. I don't know how long eelgrass has been around, but oysters have been around for 1 million years. So, clearly they've had an interaction of some form. We just don't know what that relationship looks like yet. And so it's an open question. Yeah.
Dr. Zacherl, thank you so much.
Sure.
little present from the aquarium for you.
Wonderful.
Please enjoy it.
Thank you.